So what DNA helicase does is it unwinds the DNA at the replication fork. So, and in general, that's what helicases do. Okay, and you might ask, well, can't DNA polymerase do this, right? Many of you have done PCR reactions, right? You don't have any helicase in your PCR reactions, and it still manages to do this. And the answer is yes. And the reason that there is a helicase, though, is that if you just relied on this occurring by DNA polymerase providing the energy for unwinding, it'd probably take about 10 times longer. And in fact, the machine we're describing today moves at about 1,000 base pairs per second. And how fast does the polymerase move in your PCR reactions? Anybody know? Per second? Per minute. Okay? 60 times slower. Okay? And that's actually after you've fully denatured the DNA, and so all you're getting rid of are little secondary structures during your synthesis, right? Because you always heat to 95, anneal your primer, and then extend with the polymerase. And during that time, everything stays generally above 50 degrees, so you're not going to get a complete annealing of the two strands. Okay, so helicase basically makes this all work much faster, and it turns out to be an organizing principle for the structure of the bacterial and probably also all eukaryotic, replication forks. Okay, so how does the helicase work? So it uses ATP to separate the DNA strands. And it does so by encircling one strand. Let's see, I'm going to go over here. So they're typically, actually, let me be careful with this. So we're describing a big set of enzymes. There are hundreds of different helicases in cells. But what I'm going to be talking about are replicative helicases, the helicases that act at replication forks. Okay? So replicative helicases, and are formed as ring-shaped hexamers. And this ring encircles one strand of DNA and displaces the second strand. And you can see that in the illustration that I've got up here. Okay, in this case, we have a helicase that's on the lagging strand template. Imagine there are 6 little balls in a circle, and those 6 little balls surround one strand, okay? And if you imagine this simply moving directionally on that single strand, for it to continue moving forward, there is not enough room, and not only that, there's a topological problem. You'd have to actually bring this 3' in and thread it through there, and you'll see in a moment there's not room to do that, but there's also be very difficult to do. Otherwise, as it moves forward, it's going to have to displace this top strand here from the bottom strand as a physical requirement. And what I'll tell you is that's exactly what it does. And in fact, remarkably, while you need to have the strand it's moving on be continually DNA, people have made artificial constructs where they'll replace like 50 base pairs of DNA with polyethylene glycol. And it doesn't care at all. Couldn't care less what it's displacing. Okay? Just goes right on through that, and after it gets through displacing that, when it reaches the other DNA again, it keeps on going. So what's going outside the ring is of very little interest to the helicase. What's going through the ring has to be DNA. Okay? It's recognizing the DNA there. Okay? So that outside is basically just being displaced, whatever it is. The inside is being contacted. How's it doing that? So this is the structure of a viral replicative helicase.
It's a homohexameric complex, so there are 6 identical components, and here, each of these components is shown in a different color. This particular structure is very informative because the researchers happen to have caught the helicase bound to single-stranded DNA. So the gray molecule that you see extending down the middle of the helicase is single-stranded DNA. You can also see that there's not enough room in there to put in another strand of DNA. It's actually very tight, even to fit the single-stranded DNA through the central channel of the molecule. So that's the first clue as to how these helicases work. There's simply not enough room for double-stranded DNA to go through the center. And that'll come into play when we talk about how they bind DNA. The second thing I want to point out is that each of these 6 proteins is an ATP binding and hydrolyzing enzyme. The positions of the 6 ATP or ADP molecules are shown here in red. What I want to emphasize is that not all of the subunits are going to be ATP bound at any point. Not all of them are going to be ADP bound at any point, and not all of them will be bound to no nucleotide at any point. And in fact, from this structure, it seems that they move in a sort of stepwise fashion between those states, although that could be a crystallographic artifact, so we can't say that for sure. Inside, there are these hairpin structures that reach out and touch the DNA. If we strip away most of the structure here so we can focus on the interactions between one of these 6 subunits and the DNA, we can see this much better. This hairpin actually interacts with individual bases, and the way these enzymes work is more or less like paddling down the DNA. So this is what the interaction looks like when the yellow subunit is bound to ATP. We can see that the hairpin is interacting with this blue base. When it hydrolyzes ATP to make ADP and phosphate, it is then bound to ADP, and this causes the hairpin to move down, bringing the interacting base and thus the rest of the DNA with it. Then, when it releases the bound nucleotide, it is in this state. And you can imagine what will happen when it binds another ATP. It returns to this state to find a new nucleotide further up in the DNA. So with all 6 subunits going through this process, the helicase is able to move the DNA down with respect to the helicase.
So you can see how this would work. It binds, it hydrolyzes, it lets go, it moves up and binds again, interacts with a new base pair, okay? Just like paddling down the DNA, okay? So that's how helicases actually accomplish work in the cell. And the important thing I want to make, and we'll talk about this twice today, but it's just as important. So a lot of people think, oh, it's the ATP hydrolysis that does the really interesting work. And actually, there is some interesting work here in that this changes position when it hydrolyzes ATP. But it's just as interesting what happens to this hairpin when it binds ATP. It's moving actually farther when it binds ATP than it does when it hydrolyzes ATP. And it's important to understand that when an enzyme uses ATP, the important part of the reaction may just as likely be the initial binding step that causes an energetic change as it is the hydrolysis step. Okay? So that's the idea of how these enzymes work. We know this fairly well for this enzyme. There are many others like this, and there are suppositions that similar things are going on in those enzymes as well. Okay, so that's how they function. And I'll just say that in bacteria, this is sort of an interesting difference. The helicase encircles the lagging strand template. Okay, and in eukaryotes, the helicase encircles the leading strand template. And you might want to think about what that means about how they function as we talk about the rest of the properties of helicases.